Thank you so much for the introduction and the invitation. It is such a pleasure to be here to talk to the audience, especially the young generation in the audience. You see scientists on the podium talking about discoveries and inventions. You read about their accomplishments in their resumes. But what I wanted to do today was to tell you about my journey, tell you about what we do, but more importantly, how did we get to doing what we do? So, like many of you in the audience, my journey started at a simple town, Solapur. That's where I'm from. I was born there. And I went to this school. This is a picture of my school. And among the many things in school, I got amazing friends. So, for those of you who are still in high school and looking around you, friendship with your peers is going to be one of the most significant assets that you're going to get there. But when I went to school, what you see, those of you can read Marathi, you'll see the motto of my school, Hati Ghalte Tadisnya, which basically means finish what you start. And that was one of the most significant lessons that I learned in my life. It's very easy to think about many ideas. Ideation is doable, but the execution of the ideas is very difficult. And that's the lesson that I've taken throughout my journey. And even today, that's something we really cherish very actively. So when the time came for me to think about my next step after high school, I was really trying to figure out, what do I want to do? What do I want to study? And in that question, or in answering that question, my teachers were very instrumental. This teacher in particular, his name is Keshav Shinde. He was a high school teacher. So I learned two things from him. The first is that he was the teacher who told me that a teacher is not somebody who stands on the podium and talks at the students, but really mingles with the student, sits with them, and works with them. And that really inspired me to become a teacher, and that's why I became a professor. But the second thing that he really taught me is to go to a school called UDCT for my higher education. And you have to remember, back in Solapur, this was in the late 80s, before the internet days, not a lot was known about the outside world. And it was really his advice that I took to go to a school, UDCT, which is now known as ICT. And that really opened my world. Uh, Solapur was somewhat disconnected from the world before the internet days, but UDCT had people who were directly connected to the outside world. They really opened up the world to me, and I saw the world through their eyes. And among them were some of the most prominent professors. Professor M.M. Sharma, those of you in yesterday's session, you heard his name, Professor G.D. Yadav, and Professor Bhagwat, who's in the audience today. He's the director of IISER. And these individuals, this picture, by the way, was taken at the 25th anniversary of our graduation about 5 years ago. And these individuals really taught us the principles of chemical engineering. Now, I don't know how much you know about chemical engineering. When I started at UDCT, I did not know much. And chemical engineering really was designed to teach us how to build reactors or how to build plants which can produce useful things for the society. Things like petrochemicals, things like paints, things like plastic. And to do that, you had to learn how to design reactors, how to design separation columns, etc. And I thought that when I was going to graduate, I was going to work at a plant like this. What you see in front of you is a picture of a chemical plant. It produces petrochemicals, it produces oil and other useful chemicals that we use in our everyday life. And when you look at it, it is really amazing. And what is really most amazing about it is the scale at which it operates. Now, in this particular plant, liquids are moving here and there. Millions of tons of material is being transported from point A to point B. And what is remarkable about that is that we don't fully understand everything about how liquids move. Now, those of you who are fans of physics, you may appreciate this. If I give you a cup of tea, and asked to start running with it, and then tell me exactly how does the tea move in that cup. You can't tell that to me because it is not easy to predict the motion of fluids with a very high precision because we don't fully understand it. Now, how is it that we don't understand the motion in the cup of tea, but we are producing millions of tons of materials by making the liquids move? And that's where I learned what chemical engineering really is. So here is the summary of chemical engineering that I learned in 3 sentences. Understand what you can, approximate what you don't understand, and appreciate how things are connected. So what I mean by that is that when you see complex things around you and you wonder exactly what's going on, you may not fully understand everything, but do make an effort to understand whatever you can from what you see. But be fully aware that you're not going to understand everything that you see. So in those cases, make approximations, understand correlations, develop correlations to make as much sense out of it as possible. And then appreciate that things are connected. Nothing works in isolation. We are a system of networks and connectivity. So do appreciate how things are connected. And when you follow these 3 principles, understand what you can, approximate what you don't understand, and appreciate how things are connected, you can solve very complex problems, even if you don't understand everything about it. And that pretty much has become the philosophy because everything that we look around is a large complex problem. And you can approach those problems with these 3 principles. So when I graduated from UDCT, I wanted to do my PhD. And I looked at many places and I ended up at this place. This is Massachusetts Institute of Technology, MIT in Cambridge. And I came here thinking that I'm going to work in some sort of reaction engineering chemical plant area. Maybe I'll try to understand how to develop a new reactor, maybe build a new reactor. But coming to MIT was a complete transformation in my approach to science. There, I met this individual. You may have seen his picture. He is Professor Robert Langer. Robert Langer is one of the most noted chemical engineers today. He has been responsible for many discoveries, many inventions, and he's an entrepreneur by himself. And he may be most known to you as the founder of the company Moderna, which made the mRNA vaccines during COVID pandemic. And when I met Bob, I was fascinated by the fact that he was looking to solve biological problems using chemical engineering. And that was a revelation. What does engineering have to do with biology? When I was studying engineering, I was told not to study biology. In fact, the last class I took in biology was in the 10th grade. But then I remember chemical engineering is really about solving complex problems. Understand what you can, approximate what you don't understand, and see how things are connected. And human body is probably one of the most complex systems that you will deal with. So figured, well, why not? Why not use chemical engineering to solve biological problems in the human body? So I started working on a project trying to make needles obsolete. Now, many of you may know, some of you might have experienced that medicines are quite a few of them are given by injections. For example, insulin for treating diabetes, vaccines for treating or for preventing COVID and other infectious diseases, they're all given by injections. And nobody likes injections, right? They are painful. Uh, you don't like the sight of needle piercing the skin. So I started wondering, is there a way to deliver medicines without using injections? Why not just put the medicine on the skin as a patch and let that enter into the body? That will be non-invasive, that will be painless, and the patients would love it. The challenge is that you can put the drug on the skin, but it cannot go into the body from there. The job of the skin is to keep things out. So it doesn't want anything going into the body. So we had to do something to open the skin up. And for that, I looked to sound waves. I wondered, can we use sound waves to open the skin up? Now, these are not ordinary sound waves, these are ultrasound waves. They work at a frequency much higher than what you can hear. And we eventually did develop a technology that uses sound waves to open the skin up, and then you can put a patch, and insulin can go into the body for the treatment of diabetes. We could also deliver vaccines that way. So that was interesting. We published this research in a journal called Science, which is one of the most prestigious journals, very difficult to publish in. And they were excited that we have a new way of delivering medicines. So we published that. And within a few hours of paper coming out, there were news stories all over the media. The New York Times, USA Today, Wall Street Journal, all reported that there is a new way of avoiding injections. I was giving interviews to BBC and Discovery Channel talking about this discovery and invention. And that completely changed my mind. Up until then, research was a means to do a PhD for me. And I suddenly realized that it can have a deep impact on medicine. And you have to realize there was a big change. A few years ago, I was busy playing cricket with friends over the weekend. That was the thing to do. And then suddenly, we had this platform to demonstrate a new treatment, a new way of delivering medicines to patients. Diabetic patients started calling me. When will this method be available in the market? When can they use it? How can they use it? And that's when I thought, oh, we have to get it out. We have to really make an effort to get the technology into the hands of the patients. What we do in the lab cannot just stay in the lab. It has to reach the patients. So as an academician, I started calling companies, established biotech companies, asking them if they can license the technology and make a product based on that. And the typical answer we would get is that it's interesting, but it's too early. It's a research stage technology, not a commercial ready technology. And we tried quite a few of them. So when they felt that it's too early, the question was, what do we do? And I figured if it is really important for me to take the technology out in the patients, we got to do it ourselves. So I started a company. I became the co-founder of the company. I became the employee of the company with the sole objective of converting my academic invention into a commercial product. And that was a new thing then. This was 30 years ago when academicians did not really leave their lab to start a company. So it was not quite easy to do, but we did that. We were able to raise money from the investors. We were able to get clinicians interested. And as an entrepreneur, I was handling all kinds of interesting things, from deciding company's policy and research direction all the way to cleaning things when necessary. That's what entrepreneurs do. And we did succeed. Over a few years of research and development, we developed the technology. We took it from a research level device into a clinical technology. The device was called SonoPrep. The device was approved by the FDA for use in patients. And in 2003, it received the Popular Science Most Innovative Medical Product of the Year. So that was remarkably satisfying to see, to see a technology go from the lab into the hands of the patient. But deep down, I wanted to figure out what is it that I want to do next. And I like commercialization. I like the idea, or I like the process of translation and commercialization. But I was more excited about the whole cycle of discovery, demonstration, and commercialization. And that can be done in academia. So I wanted to go back to academia to redo this process multiple times and demonstrate the potency, demonstrate the power of invention and translation. Now that turned out to be quite difficult to do because nobody wanted to give me a job, right? At that time, this was 30 years ago, there was a clear uh, preconceived notions as to what academicians should do. Fundamental science was really driving a lot of academia. Applied science was really not looked upon very favorably. If you wanted to get a faculty job in the US, the thing to do was do a postdoctoral research and then apply for a job. And here I was openly looking to do applied research. I had started a company and I had experience in industry, and the fit of such person in academia wasn't clear. But one university offered me a job. It's the University of California, Santa Barbara, a beautiful place on the West Coast in the US, in the city of Santa Barbara. And I started as a faculty there in the late 90s. And I was trying to think as to what will we do in research, what will drive our process and thinking about research. And at that time, I was really fascinated by the process of drug development. You take any drug, drug that treats cancer, diabetes, infectious disease, autoimmune disease, and if you look at the cost of developing a new drug, currently it's about $2 billion, that billion with a B, and it takes about 10 years to develop a new drug. Now think about that for a moment. I mean, $1 billion, you can look at it from different perspective, but just to provide one perspective, take a look at this picture. This is Mars Curiosity rover. This thing took off from the planet Earth. It landed on Mars. It moved around. It took samples. It took pictures. And the whole thing costed less than $1 billion. And those of you are following India's space mission, it wasn't even a fraction of a cost. So it is more expensive to deliver something into your own body than it is to send something far away to a different planet. Why is that? That process really started bothering me. That's this dichotomy of the complexity of drug development. And fundamentally, I'll tell you a reason why it is so difficult and so complex. If you take any drug product, any pill, that next time you take a pill, look at it, or any injection or any drug that you use, it has two components. The first component is the actual molecule that is the drug. And the second is the supporting technology that makes sure that the drug can go to the place in the body where it needs to go. If the drug cannot reach its desired location in the body, it's not going to do its job. In fact, it's going to cause more problems, right? Imagine this. If you take a drug and goes everywhere and the drug has some toxicity, then the whole body is exposed to the toxic effects, like in chemotherapy. And if it cannot go to the right location, it won't be effective, so it will lose its efficacy. So the way the drug companies typically deal with that is because it's very difficult to control the transportation of drug in the body, because the body is not designed to carry drug, right? Medicines are relatively new. Human body is evolved, long time ago, and it's not designed to carry drug. It's designed to protect itself and do its metabolic function. So when you put a drug in the body, you have no control over it. So we wanted to establish a field that can allow you to separate the side effects of the drug, and that field is drug delivery. And what it does basically, it allows you to control the destination of the body better, in the destination of the drug better in the body, so that you can more effective drug therapy. Now, fundamentally, why is it so difficult to control the destination of the drug? It comes to body's own biological barriers. As I mentioned, body is designed to control its own metabolism, not to carry the drugs. So skin, for example, you put the drug on the skin, it doesn't want to go in. You swallow a protein like insulin, it doesn't want to go in. It gets digested in the stomach. That's how it digests food. Even inside the body, you cannot control the destination. It is very difficult to take the drug into the brain. So how do you train brain disease? The numbers you see here are the efficiencies, the natural efficiencies with which drugs can cross these organs. These are extremely low numbers, 0.1% and 1%. As an engineer, I don't find them acceptable, right? If you take a taxi or if you take a plane, and if I tell you that only 1% of the planes land, you would never take a plane. Or if I tell you that only 1% of the emails reach the intended destination, you would never send an email. So we need high efficiencies of transport so as to control the drug distribution in the body. So we started a research program to improve the efficiencies with which we can control where the drug goes in the body. So we developed a pill that can deliver insulin orally using a technology called ionic liquids. So here is a picture of me holding a pill, uh, and the pill can go into the stomach. It protects insulin and enhances absorption into the body. In another strategy, we started using body's own cells to control the distribution. Right. So here, what you see are the red blood cells of the body in red, and the blue dots are the drugs. So by putting the drugs on the surface of the red blood cell, we can control their distribution in the body. We can make them go into the brain for the treatment of brain diseases. Another example is putting drugs on body's immune cells. As I mentioned, many organs in the body are not accessible to drugs, but immune cells, our own immune cells, can go wherever they want. They have free access. So why not just use them to hitch a ride? So we developed a strategy that we call backpacking, which is this purple patch here. We put that on the immune cell and let the immune cell deliver it into the sites of the tumor and autoimmune indication for the treatment of this disease. So with these strategies, we improve the efficiencies dramatically. So now we are operating at 10% and 20% and 30%. So that's a dramatic improvement. We are not stopping here. Our goal is to get as close to 100% as possible so that we can control where does the drug go in the body and make more effective drugs and make them in a cost-effective manner so as to reduce the burden of drug development on the society. Now, while we develop the technologies, I wanted to sort of go back to the point about entrepreneurship and translation, and we wanted to make sure that these technologies reach in the hands of the patients. And as a process of this, not only we wanted to do it about own technology, but also wanted to promote universities taking an active role in commercialization. So as a part of this, a couple of years ago, I wrote a report with the National Academies. And the main point of this report was, as an academician, when you do research, you train students, which is a wonderful thing. PhDs and masters are a wonderful outcome of academic institutions. There are publications which come out which are very important. But along with that come a lot of patents. So in the US, universities collectively produce about 6,000 patents a year. And many of these patents have the potential to make a new product, but they don't. So there is a tremendous opportunity for the universities to benefit from that. So in our own experience, we have tried to do that actively. We have started a number of companies following the same principle of ideation and translation. Some of them I'll be happy to talk about if you have any questions. But one in particular, let me call out a company called 7 Sense Biosystems, which is now called Your Bio. And here we wanted to see if we can develop a technology which can fundamentally change how blood is collected. Right. So those of you who have taken a blood test, you go to the clinic, you sit in the chair, and the needle goes in and takes the blood out. It's a very cumbersome, very painful process. Wouldn't it be nice if we can collect the blood from the patients in a non-invasive and painless way? So we developed a device that comes in mail to the patient by post, and you put that device on the skin, you push a button, and it collects a small amount of blood without patient feeling it and without patient seeing it, and the device is shipped back to the lab for the test. So instead of you going to the clinic, the device comes to you. So this can potentially transform how diagnosis is performed and can address a very important societal problem. So I'll sort of conclude my talk by kind of sharing with you some of the uh, lessons that I have learned through my journey. As I mentioned, we have gone from sort of engineering to biomedical engineering to discoveries to translation and commercialization. And as a part of this journey, I have come across many, many institutions, many, many individuals, and through that, we try to distill what have we learned that we think can be a good founding principle for the journey ahead. And the first thing I want to call out, and this is especially meant for the young generation out here in the audience, is to be curious. Curiosity drives research and science. Every discovery has started by somebody asking a good question. A good question that can come from any part of life. There are no bad questions, but uh, unless a question is asked, the journey doesn't start. So every time you see an opportunity to question things around you and sort of be sort of exhibit your curiosity around you, that's going to be the first important step of the journey. Second is to be bold and dream big. Right? Dreams is what gets things done. So while you are dreaming, why dream small? Think big. Try to solve a problem in a way that nobody has solved before. Try to take on a problem that is really bothering the society. And these big dreams will eventually get converted to reality. But the big part is an important part of it. Don't be afraid of failures. Now, I told you about many success stories. If I tell you about our failures, that will be a much longer presentation. Many things fail before something works. And that's an important part of the journey. Failures is a way of learning. If everything that you propose works, that probably means you are not asking a tough enough question. So failure is a sign that you're asking important, difficult questions. So don't be afraid of failures. Keep pushing them. Keep pushing your journey further despite failures. Surround yourselves with great people. This has been my experience. I was fortunate to be surrounded with great people all throughout, from my school to UDCT to my PhD even today. And it's important because you cannot always see everything by yourself. You have to sometimes see the world through the eyes of people who have seen it before. And great people are magnet for more great people. And eventually, science is people's game. People get things done. So make sure you surround yourself with great people. That's a guaranteed recipe of some sort of success in your journey. Be persistent. Success comes in small incremental steps. Now, there is a perception in science that there is a eureka moment. Nothing works, and suddenly one fine day everything works, and you come out saying eureka. That's a myth. In reality, that's not how science progresses. Every day there is small progress, and when you compile progress day after day, year after year, eventually it adds up to something quite promising and quite impressive. So don't look down on small incremental steps. Eventually they add to something quite substantial. And finally, be humble. Humility is one of the most underrated assets in science and life in general. Uh, Humility does not mean don't talk about what you've accomplished, but it basically means to keep yourself grounded. Realize that for every question that you answer, there are probably 10 that haven't been answered. And looking at the world from this angle of humility is a very likely route to find something significant, especially asking important questions. So I would strongly encourage the young generation here to follow the path of humility in the pursuit of science. So my time is up. So let me stop here. Thank you again all for coming, and I'll be happy to answer any questions. Yes.
So I'm Shreyas, and thank you for the wonderful talk you gave. I have a doubt, like you said, you have seventh sense biosystem, which can detect the blood, or the blood test is made easy with non-invasive. So have you been working on some of the ways to detect the glucose? As for the diabetic patient, it is hard to again and again take the blood samples for the glucose. So do you have any insights about that?
Absolutely. So that's so measuring glucose actually is how we started the journey. We developed a technology where you can put a patch on the skin and will continuously measure glucose that comes out. And in a diabetic patient, as you know, measure glucose level multiple times a day, every time with a finger stick. And wouldn't it be nice if there is a continuous measurement so that you can get a readout as to where the patient is going? So that technology was developed and was tested in the clinic. The device that I showed you for blood collection is specifically meant for tests where you need the blood. And there are many examples where whole blood is needed. So it's intended for that. But in general, I think the field is and should be headed towards strategies where diagnosis becomes easier and easier. So right now there is a world of medical technology, there is a life, and the interface between the two is not very easy. Just like how we are used to, using phones and other devices because they make an easy interface with our life. Diagnostic tests should do that, and that will improve the frequency of diagnosis, and that will increase the likelihood of early diagnosis of major diseases. And early diagnosis means higher rate of treatment. And
So I had a quick question. Firstly, congratulations on what you've done. To be able to see someone who's taken something out of academia and actually got it commercialized is it's quite rare and very relevant for this audience here because there's a lot of people here, budding entrepreneurs, who are doing it from the underlying science. One question that I had for you, if you were to look at that, uh, the insulin delivery without the needle, that technology, if you were to look at it when you developed it in the lab and as it came out into the open, what do you think the final outcome was directed by? Was it only the science? Was it the venture capital? Was it the companies that might have been interested in it? How does that science get directed? I think that's particularly important for entrepreneurs to hear.
absolutely, that's a great question. So when things are happening in the lab, you can fully control it because it's within your realm of control of science. Once you take it out, other factors do influence. Funding is the first one. The idea has to be funded by somebody. Developing medical technology is expensive, so you have to have early stage investors who are willing to support that. The second is the payers. Whoever is going to pay for the technology when the time comes to use. There are some technologies where patients themselves pay, in which case you can communicate with the patients and they can tell you. In some cases, the insurance companies pay and they will dictate what technologies get into the commercial world. But either which way, that question has to be answered. So what I have found is that as long as the patients really want it and it addresses an unmet need, it is possible to align all the people along the way. It may take time. It won't happen all at the same time. So you have to show progress to subsequently sequentially engage people. But that essentially is the role of the scientist entrepreneur to make sure that all the things are aligned and the technologies don't fall through the crack in between. And that does happen, by the way. Of all the academic technologies, only a small fraction eventually make it to the clinic or the market because somewhere along the way the message gets lost and there are no supporters of the technology.
Yeah, so first of all, congratulations and really appreciate it's a great innovation. My question is to you as a human being. So you have been in academics and entrepreneur. So how did you strike balance between these? Because the kind of challenges maybe are definitely different. And as a human being, how what is your guidance or to the new generation? How to strike the right balance between these two, academics and then entrepreneurship? Yeah.
No, that's an important question. I think the first thing I would say, the most important thing is to have something of high significance that you're pursuing. Dream big. Whenever the ideas are big, it is easier to navigate and get people aligned to make things happen. So in the lab, if the idea is big, it is easier to recruit people to help you with that. And when the idea is in the lab, it's all about science, right? So at some point, you had to decouple the ultimate goal from what you're doing at the moment. So in the lab, the focus is entirely on science. Can we solve the problem in an effective way? Is our solution better than what's out there? Are we really making a difference? Once that question is answered, we're kind of putting a different hat and figuring out how to take the idea forward as an entrepreneur. And I must tell you that there are some potential tension between the two. In the lab, when we don't have any constraints of cost and the complexity, we can develop whatever technology we want. And sometimes academia looks at complexity as an asset, right? If the idea is complex, it makes a good paper. The same complexity, you try to put that in the device, it becomes a liability because they become failure points. So every time we take the technology from the lab into the commercial world, we take it apart and rebuild it with the goal of simplifying it to help translation and commercialization. So like anything else, you had to be really thinking about what hat you're wearing at that time and make sure that you're following the principles of that hat so that you guarantee the translation of idea all the way from ideation to commercialization.
Sir, thank you for a great talk. And I'm, sort of especially, sort of grateful for that thinking framework you mentioned. Understand what you can, approximate what you don't understand, and appreciate how things are connected. So, if you could share one personal example of, first principles and correlations and approximations and systems approach which you found relevant from your own life, sir. Thank you very much.
Sure. I'll give a technical example. So the drug delivery product that we developed for delivering across the skin, that was an example of that. When it comes to transport of drugs across the skin, we could actually understand it quite well because it's a process of diffusion, which is fairly well understood in the world of transport phenomena. So we can predict from first principles how drugs move across the skin. Now, once they cross the skin, it's very difficult to predict what happens because there is blood circulation, there is subcutaneous tissue that starts absorbing things. So we had to approximate and develop correlations to connect the transport from the skin into the body. Once the drug goes into the blood, the liver and the kidney start actively removing it. The liver digests it, kidney filters it. So the connectivity of the tissues and the blood circulation becomes very important. So it really took a systems approach of first principle based diffusion across the skin to correlation to understand absorption from the skin and clearance of the drug based on the connectivity of the organs. That got us from the start to the finish in developing that technology.
Good afternoon, sir. This is Krish. So, really love the framework and the learnings because that is what takes us to the outcome. So, my question is that you have been working on the efficiency of drug delivery. Where do you see the role of nanotechnology coming in, and how long will it take to mature that?
So nanotechnology is a big part of it, Krish. So there are multiple technologies that are being used to solve the problem. Nanotechnology being one, CRISPR another, and there are many such technologies. So the picture I showed you of red blood cells, those blue dots were nanoparticles that were sitting on the red blood cell. Now I must say that developing the nanotechnology in itself doesn't completely solve the problem. You still have to figure out where will the nanoparticles go. If you inject nanoparticles in the blood, most of them go to the liver because liver basically is the clearance organ of the body. If you want to take them elsewhere, you have to figure out how to control their distribution, which is where the red blood cells come in. But nanotechnology, either in the form of polymers, in the form of lipids, in the form of electronics, all of them are very important tools and they will continue to be important tools in solving the problem. And the first example that really became prominent is the mRNA vaccine. Without nanoparticles, mRNA vaccines would not have been possible. Right? mRNA is a large molecule, it is not very stable. Unless you can compact it and make it stable, it doesn't work at all. So it took the lipid nanoparticles to compact the mRNA molecule to turn into a vaccine. So there are many examples like that which are in the works, and I suspect that you will see many more successful examples of nanotechnology in medicine in the coming year to a decade.
We have time for 2 more questions.
Thank you, Professor, for this inspiring talk. I have a very specific question, like in regards to the cellular backpacks that you showed. Um Thinking in terms of thinking in point of view of a cell, how a cell would react if some drug is put on its surface. In terms of it has a lot of receptors on its membrane, and framing it in another way, does a cell which we have coated with any drug, does it work its normal functions also, or is it just specialized for functioning or delivering that drug only? Thank you.
Now, that's a very important question. We study that extensively. So one of the requirement of the backpacks was that the backpack should not bother the cell. It should continue its metabolic function normally. So what happens is that when you put the backpack on an immune cell, a macrophage, a T cell, an NK cell, it wants to internalize it, so it's somewhat activated, but it is able to continue its normal function of translocation. It can go through the blood vessel, it can go into the tissues. And in many cases, we actually found that the backpack helps the cell because cells like support. That's what they seek. And the backpacks give them the support that they normally don't have. So in many ways, backpack not only not hinder the cell, it actually benefits the cell in terms of carrying out its function.
Hello, sir. I am Ronak, and I want to firstly say that I have seen these technologies many times, but honestly, in practical use, I haven't seen anyone using it in at least in India. A lot of people still use those needles. So, in terms of these advanced technologies, how do we make sure that after they are developed, they are tested, how do they reach the masses quicker? Do you think there is a startup possibility there also to make sure these technologies or medicines reach masses faster? Thank you.
Absolutely, and that's a very important point. Technologies have to reach the society at a large scale to make an impact. And what typically happens is that most new technologies, they make an impact wherever they are developed. That's like the first step. And once it is demonstrated, they kind of spread elsewhere. And that spread depends a lot on socio-economic factors, including the complexity of making and the cost. And probably, I would say the best example you saw was in the case of COVID, when the need was high. Even though the cost was high as well, there were ways to figure out how to reduce the cost and spread it quickly. So that does demonstrate that when the need is high, technology can spread quickly. And I would like to see more and more technologies reach the broader audience that way. But the speed with which they can spread is going to depend on the technology and the factors which drive the technology, both the patient need as well as the cost associated with it.
I am writer Ritesh Aire. My one question to develop of corona vaccine, the most world popular company name.
Sorry, can you repeat the question?
You were give me the answer of the most popular corona vaccine developed company name.
So there are many companies that develop the vaccine, COVID vaccine. Some of them develop what I call antigenic vaccines. Some of them develop mRNA vaccines. So there are quite a few, and they made impact in different parts of the world. So happy to tell you about those companies and talk to you. Maybe after, we can take that offline and talk one-to-one. So thank you very much. Time is up, but I'm available to meet and answer any questions after the talk. Thanks again for coming.
Let's have a huge round of applause for Professor Samir Mitragotri.